This program is brought to you by Emory University.
All right. Good afternoon, everyone. Thank you for being here in person, for those of you who are here, and thanks to those who have signed on Zoom. It is truly my privilege today to introduce someone who really needs no introduction, Dr. Dean Jones, who's a professor of medicine here in pulmonary critical care and adjunct professor in biochemistry. Dr. Jones received his undergraduate degree in chemistry at University of Illinois and a PhD in biochemistry at Oregon Health University in Portland. His research, as we all know, really focuses on studies related to redox biology and the interaction with metabolism. And we were just chatting earlier that his work really spans health and disease, and every disease can be linked to metabolomics and how the metabolome responds. He's really developed an Exposome research program here that is internationally and nationally known. He's published, 400, 500 and over manuscripts, and has had many leadership roles at Emory, including the program director of the PhD program in nutritional health sciences, core lab director of the Emory GCRC, executive committee of the Emory Georgia Tech Predictive Health Initiative, and has received many, many numerous awards. I'll highlight a few, the ones that he's received at Emory: the Albert E. Levy Research Award at Emory, and R. Wayne Alexander Career Achievement Award, also the Society of Toxicology Mechanisms Lifetime Achievement Award, and many others. I figured I will stop there because otherwise I'll be cutting into his time. And I can't wait to hear what he has to teach us today about Exposome. Is that the title? Okay.
Go to the first slide. Yes, the title. Yes, Exposome medicine. So yes, this is a very interesting time for a seminar. It's actually nap time. So hopefully I will can help you get a nap. So what I'm going to talk about is what I consider really a grand challenge. When I started doing metabolomics almost 20 years ago, I thought it was going to be a simple task. And the reality is it has not been at all. And it has taken me farther and farther away from metabolism and into the exposome. So what I'm going to talk about today is to just relay the concepts of the exposome and the opportunity that's going to be available probably for the next 15 to 20 years to really advance this into clinical practice, and then go through some of the more general methodologies, but not any details at all. So what are the 3 points that I'm going to cover? I'm going to start with oxidative stress because I spent half of my career on oxidative stress. And then, of course, all of the free radical scavenging antioxidant trials failed to provide health benefit. And so that really turned everything upside down, and we've transformed into redox biology in a different direction. And I'll show how that leads us directly to the exposome and what the vision that I have for exposome medicine. So this is all hypothetical at present. I have only used the term exposome medicine a couple of times in publications and haven't actually followed through with writing out a description of that. But to go back to my training, I was trained by, among others, Al Leninger on biochemistry, and he really believed that there was a molecular logic to living organisms. And so in thinking about the molecular logic, the genetic code, of course, we understand is providing us the information for storage and transmission of information. And we know that central dogma connects that information to our proteome. And this is very important. It's fundamental to living systems. But the logic for the other characteristics of living systems is not addressed by these fundamental descriptions. And so the question is, what are the principles that underlie complex multicellular development? And this is where I really want to start, because it's this is where now almost a decade ago, Helmut Cease and I introduced a set of principles that we termed the redox code, which was really intended to describe the redox organization. So that connects, the redox organization connects energetics, biological energetics, with molecular organization, that is the metabolome, and connects that to the proteome and the function of all of our macromolecular structures, and then finally the adaptation of an organism to the environment. So the key background here is that all of this evolution occurred upon the introduction of oxygen into the atmosphere. So indeed, everything about the multicellular animal life is really driven by that redox environment, that change from a relatively reducing environment to a relatively oxidizing environment. And so what Helmut and I put together was a set of principles that really to describe this. And I'm not going to go through the details. We've published many papers on this, but this concept that there was an evolved ability to sense, respond, and control oxygen that led to this elaboration of a redox signaling system. And so this is an aspect of all cellular, all macromolecular, all multicellular life, animal life. So the way we think about it is that the genetic code and the histone codes are static and they are conserved and intrinsic, but the epigenetic code and the redox code really allow for the differential expression of proteins from the same genome in different contexts. And this gives us the ability to have different cell types to respond to different conditions. And so this is really part of that elaboration of how living systems have, this is the logic of how the systems have evolved. I'm not going to go through any of the details on that, just to bring you to the fourth principle. And that is the fourth principle, and this was the first three principles when Helmut and I outlined this were largely taken directly from the literature. These were just summarized from the literature. But this fourth principle was that the evolution had really given us an integrated redox system to adapt to the environment. And so this was more hypothetical at that time, and it is what we now know as the exposome, the cumulative lifelong exposures and biological responses. And so this is indeed a Herculean task to even think about this, because it's so broad in concept. But the exposome was defined almost 20 years ago now, and we worked on this, and I honestly thought it was going to be easy, but was completely mistaken on that behalf. The main point of the exposome is that as we look at the causes of disease, heritability accounts for only a small fraction. It's really our environment and our interactions of our environment with our genome that really is driving disease. And so if we look here in the study of monozygotic twins, the only cardiovascular disease they have on here is coronary heart disease death. But what you see is it's only about 20% of the attributable fraction is genetics. And so it really emphasizes that most human disease is in fact mostly environmental. And so there are many major causes. These have been mapped out in general, things that we know about, such as air pollution, smoking, diet, alcohol, and other factors. But this is the big factor. This is the big component. The unknown is what we're really interested in terms of the exposome, because each of us has our own exposures, and unfortunately, we don't know which of those exposures are actually contributing to which disease processes. And so that is the grand challenge, is to figure out how to address that problem. So we started now, this is over a decade ago, we had a review article already on metabolomics. I thought metabolomics was going to be highly informative. It's turned out that metabolomics has been far too confusing and uninformative most of the time. And the reason for that is that we have central biological metabolic systems, and they respond to everything. And so the patterns are clearly, there are patterns that one can identify, but so far those patterns haven't really been very useful in terms of interventions. So that's cold, cruel reality. We still do a heck of a lot of metabolomics because it gives us mechanistic information, but it has not been as useful as we had hoped in terms of looking at medical interventions. What we learned already at that time was that when we did an untargeted metabolomics analysis, we got far more information than we would have ever guessed, because not only did it capture information on the metabolome, but it also captured information on other dietary components that aren't nutrients, but that are present in our diet. It provides us information on our microbiome, specific metabolites that are produced by our intestinal bacteria. It provides us information on dietary supplements and on pharmaceuticals and drug metabolites. It provides us information on a range of commercial products, things such as hand creams, things that we shampoos, and all of the things that we are exposed to, as well as environmental chemicals, many of the persistent environmental chemicals. So what we've done over the past several years is we have pushed forward with analytic capabilities. We can now measure over 4,500 environmental chemicals with the gas chromatography method. And these cover a very broad range of pesticides and different types of insecticides, fungicides, and things that are in our diet, things that are in our water, things that are in the fabrics and just that we're exposed to. And we have, in addition to those, we have capabilities now for tens of thousands of molecules measured by liquid chromatography. So this is in addition to the known metabolites, a few hundred known metabolites, we're now being able to capture close to 70,000 curated metabolites. So the coverage is incredible, but that is really part of the daunting aspect of this. We have so many exposures, and now as we're looking at individual human samples, we're seeing that the exposures are dramatically different. And therefore, if we're going to be able to understand for precision medicine, if we're going to be able to understand our individual exposures, we're going to need to have capabilities for routine measurement and evaluation of these. So in terms of the methods development, now nearly a decade ago, we arrived at this paradigm that within a single measurement, we can obtain information not only on environmental exposures, but also on biological response to the exposures. And at a population level, we're able to use this information now quite effectively, but being able to translate that to individual analysis and individual use in terms of clinical practice is something that is the grand challenge. It's not obvious how we're even going to move to get there. So in this paradigm, what we see is we have on the left the exposures, and again, we pick up a very broad array of exposures in our measurements, and we're measuring internal doses related to those exposures, and then we obtain biological response in terms of metabolomics. We've also done a very broad array of other omics in connection with the exposures, and then with health outcomes with a very broad array of different disease processes. And so where we're going with this and what we've been working on is we've been using model systems to develop the methodologies. And the methodologies follow directly from central dogma, and that is that if we integrate the data of the metabolome with the proteome and with the transcriptome or the epigenome, we actually can get an understanding of how they interact and what the central hubs of interactions are. And this is really then driving us forward in terms of understanding. So one of the main tools we use is termed XMWAS. This was developed by Karan Uppal when he was here at Emory a few years ago. And what this does is it does a data-driven analysis. So it's not dependent upon knowledge to do the analysis, but what one does is after obtaining information on the central networks associated with whatever exposure we want to look at, we can then map that back to either pathways in terms of functional pathways, in terms of metabolic pathways and so forth, so we can connect it back in a meaningful way for interpretation. And so this was really validated by Youngmi Go several years ago in a study of cadmium toxicity. And what it showed was that indeed you can, when you look at the most central responses to cadmium exposure, among those central responses are oxidation of mitochondrial proteins involved in fatty acid metabolism. And then when one looks at what metabolites are actually changing with the oxidation of those proteins, what one finds is that they are fatty acid metabolites. And so then, of course, with model systems, it's possible to go back in and validate it, which she did in that study. So what that has given us is an understanding of the oxidative stress responses in biologic systems. And this is so important because what we know from our experimental studies is that the proteome controls transcription. We also know the converse is true, that transcription controls the proteome. We also know that the proteome controls the metabolome, and the metabolome controls the proteome. And so this is really it's in a certain way, it's trivial because it's obvious that it has to be this way, but it's fundamental in terms of the way we think about biological responses, because what it's telling us is that in response to every exposure, we have transcriptome changes and we have metabolome changes, and those are feeding back on each other. So it's all an integrated network. And so that's coming back to this concept. It's a network that we have to look at. And this was really hit home by a study that Jolene Fernandez did when she was a postdoc here. And this was, again, a model systems study. But what we did was we looked at the networks, community structures associated with manganese. So manganese is an essential nutrient. In this model system, we could go from a low suboptimal level of manganese up through an optimal level of manganese to a toxic level of manganese. And we could look at the biologic response. And so this is a snapshot of the biologic response. And the thing that I want to emphasize is that there's 4 communities of interactions. I'm not going to go through all 4 of those, but I just want to show you within this network structure, there are positive correlations that are occurring with regard to a change in manganese. There are additionally negative correlations that are ongoing with this change in manganese. And the remarkable thing is that they are simply mirror images of each other. And when we go in and look at the metabolite transcriptome responses up here in the upper right, what you can see is that we have some positive and some negative response structures. And so this is really emphasizing that as we think about biological responses to exposures, we have to be thinking about the opposing reactions, the opposing responses. And this is what makes it so difficult about looking at metabolomics alone, is because when we look at metabolomics alone, we don't know whether a response is a protective response or an adverse response. And that is the limitation that we have to work with as we move forward. So the holistic picture is that the transcriptome, the proteome, and the metabolome are an integrated network. And this, is obvious in a certain sense. But on the other hand, that's really points to the way we have to think about things at an individual level. We have to be thinking about the adverse responses and the beneficial responses simultaneously. And we don't have a roadmap for what's good and what's bad. And so that's a big challenge. I wanted to just give an example of how this is actually true in a human tissue. This is a study that Ryan Smith did. Ryan is a assistant professor over at the VA in the pulmonary division now. He did this when he was a postdoc with us. He looked at the metabolic associations with cadmium in the human lung. And in this case, these were mostly lungs that from end-stage lung disease. We had some healthy lungs and different disease processes. So it's really quite heterogeneous. But cadmium is a main component in cigarette smoke and highly toxic to the lung, causing lung cancer as well as a number of other lung pathologies. But anyway, there were metabolic associations associated with cadmium. There were cadmium. The way it's toxic is that it interacts with selenoproteins, and selenoproteins are antioxidants. So they protect us. Glutathione peroxidases, for instance, are selenoenzymes. So the cadmium is toxic because it binds to those and inhibits those antioxidant proteins. And so what we see is that from the data is that there's an antagonistic interaction between the cadmium and the selenium. And you see a central group of metabolites here that are changing in opposite directions to cadmium and selenium. And these turn out to be fatty acids, inflammatory fatty acids. And so what it's really showing is that in a human samples, disease samples, that in fact, that antagonistic structure is there. There's the good beneficial reactions to selenium and the harmful reactions to cadmium. So I wanted to give one more example of the integrated view, and this was a study that Ken Liu did when he was a PhD student with us on looking at microbiome metabolism. And what it emphasizes, what he discovered is that there is a chemical that is produced by our intestinal bacteria called delta valerobetane that turns out to be an inhibitor of mitochondrial beta oxidation of fatty acids and actually causes obesity in mice and correlates with obesity in humans. Anyway, this metabolite is dependent upon what we eat, produced by our bacteria, and act upon the host. And so it's really emphasizing that we're not just our host, we're not just ourselves in terms of our biologic response, but we're responding as a holobiome, an interactive unit with our microbiome. So I'm not going to go through any details on this. The main point is that the evolution has really given us very broad networks of interactions. These are the protein interactions. These are the hydrogen peroxide signaling interactions. This was a review that Helmut Cease and I had a few years ago in Nature Reviews, and it really emphasizes that these redox, what we always have thought about in oxidative stress, that now we're understanding that the reason that we saw all of those reactions to oxidative stress is that redox signaling mechanisms are so pervasive in biology. And so they're really functioning, impacting most systems that we have, most functional systems that we have. So we now have tools to be able to interrogate this on an individual level. These are not brought to the clinic yet, but this is definitely technology that is amenable to clinical use, and that is looking at mitochondrial function. This is with a seahorse assay, and what we can see is that when we look at the metabolic associations here in the circles with the functional measures of mitochondria, we see that there are different metabolic clusters, different pathways that are being associated with different characteristics of the mitochondrial function. And so this clearly will be very useful as we go down the road for looking at exposome, at the xenobiotics, the foreign chemicals, at the dietary components and their interactions, their impact upon mitochondrial function. So I'm going to come to the last part, and I did this right. I left plenty of time for discussion, so we'll be fine on this. So the way I have defined exposome medicine is really as this concept of using detailed individual exposure histories for the purpose of prevention, diagnosis, management, and cure of disease. And again, this is hypothetical at present, but this is a challenge. This is where we have to go, because if we're going to actually understand which exposures are contributing to which disease processes in which individuals, we're going to have to get here. And so I'm going to give you an example of exposome detective work. So this was just published earlier this year, and Young-Mi Go was the leader on this, and she did. I would love to take credit for it, but all of this is her work, so I will just acknowledge that up front. So what she did was used archival samples where there were known health outcomes, and so the archival samples in this case were from the Public Health Institute in California. This project was the Child Health and Development Studies was with Barbara Cohn as the director, and this was a collaboration with her and her team. And so in this cohort, they collected, as part of Kaiser Permanente back in the late 50s and early to mid 60s, they collected samples from women who were undergoing care. They were during the pregnancy, and what they did was, this was for environmental health concerns. They collected samples from the mom and from the father during the course of the pregnancy, and then archived those samples. And then what Barbara and her team have done has been to follow up on the outcomes on that cohort. And there are about 20,000 couples in that cohort. And so their children and grandchildren and now great-grandchildren have follow-up information. And so it's particularly straightforward with regard to cancer, because there is a California cancer registry. So it's possible to follow up on all the individuals who are still in the state of California would have cancer outcome. So this study, we've done studies in the past, which Barbara and her team have done studies on, showing that DDT is actually, the maternal DDT is associated with breast cancer outcome in the daughters. And so this study was outcome in the women themselves. And in this analysis, what we did was we used the high-resolution mass spectrometry and then selected signals that were positively associated with breast cancer as an outcome. So these are samples that were collected anywhere 5 to 50, 60 years before cancer. And so we're looking at what signals were present in the plasma. Actually, this was serum samples at that time, and then subsequently associated with the breast cancer outcome. And so we applied our methods, and this is the biggest challenge right here. We have capabilities to measure up to 1 million chemical signals, and almost every one of them is unidentified at present. So we've developed methods to be able to link things to outcome, but we're so far behind in terms of the chemical identification. And this is what's really limiting our ability for the precision medicine aspect. So in this analysis, then, what Youngmi did was she selected environmental chemicals that were associated with outcome, and then she used network analysis to try to find associations with metabolic networks. And so these are the results that are in the manuscript. And so we call this exposome detective work, because what we're really trying to do is we're trying to identify suspects. We cannot prove from these studies, we cannot prove anything. Okay, so we're just looking for suspect exposures and suspect metabolic response patterns. And so what one sees is that there are multiple communities of interaction. And this community one has an insecticide that has never previously been associated with cancer. Don't know that it's ever been studied, because so many of these things that don't have to have studies. The more disturbing aspect actually is a cleaning product is also in this community. And so it makes you worry, wow, what are we exposing ourselves to? And if we go to this community, there's a retinoate metabolite that has previously been identified and linked to cancer as an outcome. This is a vitamin A metabolite, but not an active vitamin A metabolite. Then over here, we have a couple of other community structures. This is a fungal metabolite, and then we don't exactly have the chemical identity for this one. And then there's a combustion product from fossil fuel combustion, very similar to known polycyclic aromatic hydrocarbons that are known carcinogens. And then, unfortunately, a food preservative. And as we go through in that detective work, what we can do then is link these communities to what are the central metabolic pathways and associations. And this one has a food-derived metabolite, another endogenous metabolite, and then a whole series of amino acids in terms of the associations. This community is principally associated with the sphingosine, triglycerides, and phosphatidylcholines, and this community is associated with inflammatory lipids. While these last two, one of them is this fungal product is really independent, but it is associated with inflammatory lipids. And then, sad to say, glucose is in this community associated here with the food preservative in this combustion product. So what it's telling us is that there are potentially different clusters of metabolism and exposures and metabolism that are linking to disease outcome. Now, my guess is that these are probably separate in different individuals, but we haven't done the analysis, the sub-network analysis to actually determine whether these are unique to different populations or these are small effects in everybody. And these are the types of things that are going to have to be studied more. The most compelling argument from this is actually right here. And this is that when we restricted the study to early onset breast cancer within 15 years of the sample being collected, what we found was that the early onset cases were associated with amino acid metabolism. And when we looked in specifically at the amino acids that were impacted there, and this is unpublished data at present, when we looked at the amino acids, these are amino acids that impact our nutrient sensing mechanisms. These go through mTOR pathways, and this is how our body regulates our nutrients. We have sensing mechanisms that control that. And the bad news is that those are ones that are already associated with the cell survival pathways in breast cancer. And so what it really is telling us is that, yes, indeed, we're probably getting a correct message here, that there are environmental exposures that are impacting our nutrient sensing mechanisms, which are mechanisms that are linked to breast cancer outcome. And so again, it's all association data at this point, but it tells us that if we can develop these methods, then we are going to be able to come back and identify exposures that are of concern and hopefully be able to manage those exposures to prevent disease. So obviously the end of that is we have to go with targeted hypothesis testing. After we come through with identification of suspects, they're just suspects. We have to come back for proof. And so that's obviously down the road. And this is why 10 years ago, I thought we'd already have answers. Obviously, that was quite naive. We're now at the level that we're beginning to see how we can potentially move forward, but we're going to have to do this with regard to all disease processes. So the different types of cardiovascular disease, we're going to have to come in individually and sort through which exposures. And these are exposures at the detailed level. These aren't eating a Mediterranean diet. These are what are the specific components that are beneficial and which are harmful in terms of those exposures. So that's the long-term vision, is that this is where we want to go. I have to say it's daunting, but that's it's and that's why I say it's grand. It's a grand challenge because it's that daunting. But what we can conclude from the data is that the oxidative stress and redox biology really point to the need for us to transition to network methods. And that's, I think, is the only way we're really going to get to the answers, is trying to understand the networks and the different components of those networks. We have the research tools. They're really they're available now to begin to link the human exposures, even these very, very complex arrays of exposures. We're getting to the point where we can now begin to link those to the biologic responses and the health outcomes. And obviously, we're going to have to develop capabilities. We may need a new generation of exposome methods to be able to take this in a routine manner for population studies, and then be able to link that so we can get a foundation to be able to develop this. So I think that's all I have to say, except to acknowledge a heck of a lot of people in terms of collaborations. And most of what I've done has been collaborative. And that's the only way we can actually move this very challenging task forward. And I will finish up with what if you remember one thing that I say, don't let your vision be limited by what you see, because if we just work with what we see, we can't take that next step to where we have to be in order to really improve the clinical care. So thank you.
The preceding program is copyrighted by Emory University.